The overall goal of the following experiment is to determine cytotoxic T-cell activity by comparing an impedance-based approach using the xCELLigence system to the chromium release assay. The first step is achieved by separating peripheral blood mononuclear cells from whole blood using phycol. Next, the PBMC are added to wells, along with peptide antigen, human recombinant IL-2, and irradiated PBMC to create antigen-specific CD8 T cells. The antigen-specific CD8 T cells are then co-cultured with tumor cells in both assays in order to cause tumor cell death. Results are obtained that show percent lysis of tumor cells using both the impedance method and the chromium release assay.
The main advantage of this technique over existing methods like the chromium release assay is that the impedance-based system collects real-time data and requires less cells that don't need any labeling.
For this protocol, blood from normal, healthy HLA-A2 positive donors provides a source of PBMCs, or peripheral blood mononuclear cells. Following the manufacturer's protocol, use phycol-PAIC Plus to separate the PBMCs from the blood. Next, into 6 well plates, load approximately 4 million PBMCs in 2 milliliters of media per well. To each well, add HLA-A2 binding peptide for a final concentration of 10 micrograms per milliliter. Culture the cells. On alternate days, to help stimulate